Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last few classes we have talked about several R packages and then we talked about one bioconductor package. So, these packages they are useful for data organization, plotting, etc. And in the last class we talked about one R package which we will use in subsequent class for connecting or accessing the ensemble database. In this class we will introduce another package which is actually very useful if you are working with flow cytometry data. So, this is what we will be talking about in this class. So, the topic of this class would be flow cytometry data analysis in our bioconductor. This is a bioconductor package that allows us to process flow cytometry data. Okay, so, this is the agenda for this class. So, we will first talk about flow cytometry data analysis and the packages that will be using, especially we will be using the flow core package which allows us to access the flow cytometry data, and then very briefly I will show you flow viz package. So, this is actually for visualization, but we do not always need to use the flow viz package. We can use our own visualization techniques that we have learned, and we can generate very interesting plots and graphics. Okay, so here are the steps that we will be talking about. And we will jump into the data processing part first. So, the first we will have to get the data. So, I will explain the data that we have that we will be processing in this class. Then we will filter that data. So, as you know, in flow cytometry data, when we do the experiment we often set up gates etc. and do filtering. So, we can do the same thing here, right. We can write our own codes and do a lot of customized filtering of that data to get to the actual observations, the real observations. As you know, flow cytometry data is very noisy and there are a lot of noise, background noise. Then you might have cellular aggregates, debris, all those things are there. So, we need to get rid of all these noise, debris, etc. and focus on single cell data, right. So, the idea of flow cytometry is to get measurements or expression or signals from single cells, right. So, individual cells. So, we can actually then use that data for various purposes. We can identify different subpopulations, heterogeneity, etc. We can study those. So, we will do the filtering so that we actually get the actual data from individual cells, and then we will actually analyze the data and we will generate some visualization. Okay. So, that is the agenda. So, let us first talk about the flow core package. So, this is a bioconductor package that we are going to use, and then we will actually process a data set in R. So, let us first now move on to the flow core package and see the package, right. So, here is the flow core package in bioconductor. Here we can use this for data processing. Here is the basic function file, right. Here you have some background, and then there are a lot of discussions, right, and all this manual is very informative, and it tells you how we can process these data sets. Okay. So, again for the constraints of time, right, we will not discuss everything here. The one function that we really need and will use for our analysis is this read.fcs. Okay. So, this is the file that we actually can use to actually read the FCS data, right, from the flow cytometer. So, we get the data in FCS format from the flow cytometer, and we can use this read.fcs, and this will be loaded as a data, right. So, that data then we can, that data frame we can then use and analyze like normal statistical analysis, etc. We can apply all those things on this data, right. So, that is that first critical step, and then of course, you can do a lot of other things here which are described in detail, right, and how actually you can process experimental metadata, etc. etc. Similarly, there is the other package which is the flow viz. Okay, this is another bioconductor package, and this is for visualization of flow cytometry data, and here also you can see the reference manual with discussions of the functions that we can use. Okay. So, for example, you can generate density plot, you can generate flow plot, etc. So, what FlowBiz you can do with FlowBiz is that you can generate these contour plots, density plots, right, showing the distribution or the density of points at different values of signals, right. So, for example, different cell size, cell complexity, or the fluorescence signals, right, whether it's green fluorescence or red fluorescence. Okay. So, there are different ways, right, and there are different functions to represent this. Okay. So, we'll keep it simple now. We'll process the data with flow core, and then we'll use our plotting functions that we know of, right, or we have already discussed. We'll use some of those, and we can generate these plotting the plots and visualizations. Okay. So, let's now move on to the data itself. So, here I have a data. Okay. So, you can see this. This is a data set for a bacterial strain, and we have collected these data sets at different time points. So, we have 0 hour data, 1 hour, 2 hour, 3 hour, 4 hour, and 5 hour. Okay. So, test 5 hour. So, you have these 6 time points, so where we have collected data. Okay. And what we want to see is how the signal changes, right. So, this is the idea we have, right, and whether there are subpopulations that are showing change in expression of certain genes over this 5 hour time period. Okay. So, we might have very similar data, right. You might be doing some treatment, or maybe you are exposing them to different conditions and you want to see how gene expression changes, right, or maybe you are working with, let's say, antibody, and then you want to see the subpopulations, etc. Okay. So, in this data we have both GFP and MJD channels. So, we will have green and red signals that will be there. Okay. So, very briefly I will just want to show you, right, the code and how do we process this data. Okay. So, let us now go into flow core in R, right. So, we will now first load the library. Library flow core, and we first need to read the files. Okay. And to read, as I said, we will use this read.fcs function. Okay. So, read.mcs function that will allow us to read these flow cytometry files. So, you can see these flow cytometry files .fcs, they end with .fcs. So, we can read them, and the command is very simple, right. We can say T0 test read.fcs. Okay, let us first see whether in the right directory or not. Yeah, we are in the right directory. So, let us now read this. So, T0 test, I just want to make sure the names are correct, because otherwise T0.test equals to read.fcs. I think 0 hour, yeah. Okay. So, this is as simple as this. So, we have to use this read.fcs function. This comes from the flow core package. Okay. So, we can now read the rest of them like this. So, we are now reading the 3 hour time point, then 4 and 5. So, okay, yeah. So the file name was not correct. So now we have the data. Okay, so let's see how the file actually looks like, and if it okay, so here is the summary of that file. Okay, and as you can see, some of you might be familiar with these parameters that we measure, but otherwise I'll just describe. Okay, so what we have is this FSCA file. This is the size, right? So we have this amplitude, and this is the height, FSCA, FSCH, and the width, FSCW. So this measures, FSC measures forward scatter, which is the cell size. Okay, and then you have SSC, A, H, and W. This is cell complexity, right? That's what it measures, side scatter. Then you have fit CA, this is the green channel. Then you have P Texas red A, this is the red channel. Then you have cyan, etc., blue channel, etc. All of these are there. But as I said, the signal is here, right? We have mostly in this green and red. Okay, so P Texas red and fit C. Okay, you might see some cyan channel signals, but that's, you will see the overlap because of the overlap, spectral overlap. Okay, so we will work with this FSC and FIT-C and P Texas red, these signals, and the idea is first is to filter the data, and then we will work with the visualization part, right. So before doing the filtering, right, if we don't want to read these FCS files every time, so we can actually write them something in a text file, normal text file, right, as data frames, right, so that our life becomes easier. So for that, this is how we will do, right. So what we get is something like this. To convert this to a normal data frame, we have to use something like this. Okay, so expression T0 dot test. So we can actually store these files, right. Right, dot table expression T0 dot test. Okay, and then we can simply say test T0 dot txt, right. So this is now stored as a data frame. Okay. Okay, so we can save these files in text format, so that next time if you want to work with the same data, maybe you want to look at certain different parameters, or you want to do the analysis in a different way, you just read these text files. Okay. The other way, you can also actually just store this here now, and you can say, right, just that we store this as a data frame. Okay, so now once we store this as a data frame, we can actually do all sorts of analysis, all kinds of analysis that we want to do. So we can store this in this way, or we can also load this from that file itself, right. Is read dot table. Okay, so we can do that as well, and let's see this, right, it looks identical. Okay, all right. So let's, for now, let's just store these as data frames. So we can apply all sorts of functions on top of them. Okay, so we are creating these data frames for all time points. Okay, and we can then use these data frames for our analysis. Okay, so we have now loaded the data, flow cytometry data, using this read.fcs function. Okay, now the next step is the filtering. Okay, as I said, one of the things probably I showed you. Let's see, filtering part. So okay, for anyone, right, or T0 or T5, you can see, for example, this summary of this data, this FSCA is saying negative numbers, giving negative values, right, minimum is negative. Similarly, for SSC is positive, but FSC is negative and FITC is negative, right. So how can cell size be negative, right? Of course, this depends on the voltage or the parameters that you set in the flow cytometer, but then which means we have to carefully look into the data first before we just plot this signal, right. Take all of FITC and all of these cells that we have, all the events, all the observations that we have to carefully first test, right, whether these observations look okay, right. So because you get cellular debris, there are a lot of background noise, we need to filter those out, okay. Otherwise that will influence the final outcome of the experiment or the results that we get from this analysis, okay. So in addition, we also have cell aggregates, right. So when we prepare the samples, we vortex etc., but we still have some aggregates in there, so we have to also do aggregate filtering, okay. For aggregate filtering, we use this FSCA by FSCH, right, ratio of FSCA by FSCH, or the ratio of SSCA by SSCH, okay. So here I will use the ratio of SSCA versus SSCH, because I see for FSCA and FSCH we have some negative values, right, FSCA especially. So that will change the relationship how this works. So I will use the SSCA versus SSCH value and see, right, whether we can separate out aggregates if we see different clusters etc., okay. So that's the analysis I will do first. So how do you do that? We can do this for any of the samples. So this is, we are using the plot xy function, right. So this is the x value. So we have to plot this SSCA versus SSCA by SSCH, okay. So this is what we will see. The other point you probably should have noticed is that the names of these columns have changed, right. This is data frame and that is what R does, right. If there is a special sign, right, there is a hyphen, that is now replaced by this dot, okay. You see this, for example, we have this SSCA was hyphen, right, SSC hyphen A, okay. Okay, so maybe there is some issue where, okay, so maybe the names, let us see, maybe we are not giving the right names, okay. Okay, maybe, oh, because the name is actually still retained, let us see if we can use that, okay. So there is some issue here, let us see. So it is not able to access these variables for whatever reason. Okay, so it is not accessing any of these variables. So maybe we have to get this data as write as a file and then maybe try loading that, okay. So what I will do is maybe just try, okay, it should work. Okay, so for whatever reason it should change, but it has not changed the column names, so and it is not able to access them either, okay. Usually they will be replaced, but they are not replaced right now. So maybe what I will do is I will write them as table, write those files and then load them from there and see if this issue is resolved, okay. Let us see, and okay, so let us first try this and see whether okay. Okay, so now I think the names have changed, and as you can see, they are now replaced by .a, and we should be able to access them. Okay, so there is perhaps some sort of transformation that is going on, and or maybe we have to store them as data frame. We have to say as data frame, and this issue might be resolved. Okay, so let's see if we can access this. Yeah, we can access them now. Otherwise, earlier it was not able to read them. So what we will do, we will continue that for all time points maybe, and okay, so we do for T0. We do for T1. Do for T2. Because these data sets are quite big, so it's taking a few seconds. So we have about 100,000 events for every file, right? So it's reading about 100,000 rows for each file. So that's why it's taking a bit of time. Okay, so we're almost done. Let's now get the T5. Okay, so this is done, right? Let's now load them here from the file data. Okay, so T2, T3, T4, and then final one is the T5. Okay, so we have loaded the data. Now what we can do is we can now do the filtering. So which for that we'll plot this, right? As I said, this is what we want to plot. Okay, and it will generate a plot showing these points, but then of course we have to kind of rescale, etc. Otherwise we'll not be able to see them much. So for rescaling, let's say we change the x scale, right? We have to also change the color of the points, right? Otherwise we'll not be able to see them very well because they are overlapping points. So we use the color equals to RGB, right? And let's see. Okay, so this is how it looks like. We can even reduce the point size a bit, right? So maybe you can make very small points so that we can see this data a bit clearly, right? We want to see these clusters of cells, right? So you can see these clusters now, and you can see that we have these clusters, okay? And what you see on top, these are actually the aggregates, okay? This is where we have the aggregate cells, and here you see the main cluster of cells are here, okay? So for that we can actually visually see this, and this number is around 500 or 1000. So let's draw a line here. So we have to separate out these cells. These are likely to be debris and some background noise, and we also have to separate out these aggregates here, right? So as you can see, these aggregates, okay? So what we'll do is we'll draw a line, right? So first draw an horizontal line. So it's about 1.5 value, right? If not, you can definitely try out different lines, and then AB line H equals to 1.5, right? Horizontal line at 1.5, line type 2, because it will generate dotted line. You can have different line types as well. Color equals to red, okay? And you see this line is added, and we can see, okay, 1.5 is the right cutoff, okay? So these are the aggregates. So we need to consider cells only below this 1.5 cutoff, right, where SSCA versus SSCH value is below 1.5, we have to consider those cells. Okay. And then we also have to exclude certain cells, right, or the debris, right, and this is around 500 or 700, right. Let us keep it 500. Now we can play around, you can zoom in a bit, right, you can reduce the scale, and let us keep it around 500, and then we have to, these are the right cells for us, right, these are the individual cells that we want to use for our plot. Okay. Okay, so we have done the preliminary filtering, right. You can actually go into much more detail, and similarly you can do for FSCA, FSCH, etc. You can see that for FSCA, if we plot, what we will see is there is an okay, so it's actually plotting, and you will see there is a cluster below 0. Okay, so you can also generate a contour plot, and you can see there will be clusters around 0 or below 1000. So those are actually mostly noise. Our cells are above 0, right. So maybe it's not clear here. We can see this a bit by changing this. Okay, so here is a cluster, right, and we want to discard this. These are not actually cells. Okay, and the main cluster is around here. If it is not clear, we can actually make a contour plot, right, where we, or a density plot where you see the cell density, and this is something that will tell you, okay, most of the cells are here, and this is actually noise. Okay, so we have looked into preliminary filtering. Of course, you can look at this more carefully, but what we will do is now we want to plot this, right. For example, one of the plots that we want to make first is we want to do the FSC versus fit CA. Okay, and we will do the plotting in the log scale. Okay, so what we are doing is we are filtering this, right, the data from, right, FSCA only for those cells where we apply these filters. Okay, so I will show you the first command. Okay, so we are moving in the log scale, all the values in log scale, because then we will collapse the scale, right, in smaller range. And then we are taking the FSCA value, but only for those, because it is a data frame, right. So we can use the functions that we like, right, and only choose those cells where we have this FSCA greater than 1000, SSCA greater than 500, and the and then these signals are positive, they should be positive, and the final ratio of this less than 1.5. As you have seen, right, below 1.5 we have single cells, above 1.5 we have the aggregates. Okay, so we have created this for FSCA, and similarly we will generate the same for fit CA, apply the same conditions, and now what we have to do, we have to plot x1 versus x2. Okay, what we can do is we can actually create a very nice density plot rather than just plotting x1 versus x2. We can do that, of course, just x1 versus x2, but we will create a very nice density plot. So for that we first create a data frame with x1 and x2 values, right. These will be our xy, and this is where we are actually creating this density plot, right. So this is actually creating the density plot, right. So using this color palette, and we are calculating this df density. Okay, sorry. Okay, so here is the density, and we can also look at x. So x is the density colors that we are seeing, the colors, and then we define the colors, right. Okay, these are the color codes in R. Please see, right, there you can also express colors in terms of color codes, and you can access many different colors using the color codes. And then you have the colors in, so we are creating this color for this data frame. So we have created the density for the data frame, and we have created the color for the data frame, right. So once we have done this, now we can do the plotting. Okay, and this is what we are going to do, right, the plotting part. Okay, so layout matrix is the, we are creating this plotting area first. Okay, this is the layout, and then we use this command Okay, so what it means, right? We'll see this what happens. Okay, so it is actually defining this for the first plot, right? We'll create two plots here. Okay, so we'll have two columns here in this same plot. Okay, so there are ways in doing this in R, right? And this is the first plot that we are creating. Okay, as you can see, this is cell size versus GFP signal, and here is the density plot. So which means red color means most of the cells are around here, right? And lighter shades means you have less number of cells. So as you go here, this is single cell, right? You have the cells here, single cells. Okay, and now we need to add the color scale as well, right? So for that we need to add the color scale on the right. So because so as you can see, this color scale shows us, right, where we have the most density. The most density is here, right, in this red region. That's the highest density. So most of the cells are around here. Okay, so we have created this. Now we can do the similar thing, exactly same thing. Instead of cell size versus GFP signal, we can do cell size versus the green GFP signal versus the M cherry signal, right. So then in that case, x1, x2 will change. Rest of the things, rest of the code will remain the same. Okay, so I'll switch off this now, and so x1, x2 will remain, will change, right. Instead of FSC and FITC, we just take FITC A and P takes as red A, and then we use the filters, all these filters that we have here, and we apply the same command, right, and very similar idea. You will see very similar plots here, right. Now we have GFP versus MG. Okay, and similarly we can define the color scale on the right. Okay, and this is how we are defining the color scales. Okay, so this part of the code, this is actually for the color scale, right, and the first part is just plotting this x2 versus s1. It's actually generating this density first, and then it's ordering this density and adding colors to those density, right. And in here, this second part, it's actually generating this color scale. Okay, so this PAR MAR is the dimensions of this, right. These dimensions are saying how big these plots should be. Okay, and the first layout command, it's saying like, okay, there should be two plots, right, n row 1, right, 1 row 2 columns, right. So it should be that should be the column, and the what should be the widths and heights of this. Okay, all right. So this is something we have done. We can of course save this and use for our analysis. So we okay, so we can save this as PDF and we can use for visualization later on in reports, etc. Okay, so with that we'll move back, right. So to the presentation, here are some references. So I have shown you only using how to use this with flow code, but similarly we can do with flow V's as well, right. We can take the results from flow code and we can apply flow V's, but we can also apply our own xy plot xy functions. And so I have shown you, I have given you a very brief glimpse of flow cytometry data analysis. Of course, there are a lot of things that you can do. For example, using flow V's, you can select cells, you can draw on top of the density plot, you can draw different shapes, and you can select cells from different regions and analyze their signals. So all those things are possible, but of course with because of time we have not managed to do that. And I have also shown you some hands-on with flow code package. Thank you.